Okay, moving on to section 13.2, and I'm thinking I'm going to try and cover section 13.3 as well. So let's start sharing the screen. Okay, so now let's actually talk about the steps that we need to take in order for translation to occur. So it can be divided into 3 steps, quite easy. You have initiation, elongation, and termination. Kind of like in transcription, you always have initiation, elongation, and then termination. All of these three steps require other factors, other proteinaceous factors or factors made out of proteins, and these are going to be called basically translation factors. You're going to have translation factors that are found that are specifically for initiation, some that are specific for elongation, and others that are specific for termination. So we're going to have things like initiation factors, elongation factors, and so on. A lot of these steps are going to require energy, right? And so that energy is actually taken by the hydrolysis of GTP. So this is another energy carrying molecule, very similar to ATP. You break down GTP into its appropriate GDP and phosphate, and that releases a lot of energy. Okay, so let's look at the initiation. So here, just the different components that you need. You're going to need your ribosomes. Like I said before, usually your ribosomes are not in their cool little snowman formation at this point. They only really assemble that at that point when we've actually started translation elongation or translation initiation / elongation. And so usually they're found dissociated. So you're going to have the small subunit floating around the cytoplasm and the large subunit floating around the cytoplasm. You also need GTP, right? Your energy, your initiation factors, elongation factors that will be useful later on. Oops, sorry. Trying to pause this to sorry. Okay, sorry. So you have your elongation factors. You're going to start needing your tRNAs, right? And then, of course, your mRNA, your charged tRNAs as well. So here, the elongation factors, as their name suggests, are going to come about later in the elongation stage. Right now, we're more focused on initiation factors. There's also a couple of things like magnesium. It's just important for these factors are basically proteins, and they require the assistance of certain minerals like magnesium. And so what these initiation factors do is just that they enhance the binding of the various components of the actual translation machinery. So in order for the ribosome, actually it's going to be the small subunit binding to the mRNA, it does so with the help of these initiation factors. Otherwise, things don't really stick properly together. So let's look at how initiation actually happens. So initiation occurs when your mRNA is being read. Remember, the mRNA binds to a part of your ribosome, actually. It's not like the two ribosomes, like the two snowman ribosomes as is usually depicted, and as actually I think I even stated in chapter 12 just to get the point across. But actually it binds only to your small subunit. And at that point, the small subunit starts reading, kind of threading through, you can see it a little bit here. It threads through the mRNA until we get to that initial mRNA codon, that AUG start codon, right? This is also what happens in bacteria, guys. So just remember that this is bacteria, but the same principles can be applied to eukaryotic ones. So the small subunit of the ribosome is going to initially bind to the mRNA via a specific sequence on the mRNA called the Shine-Dalgarno sequence. Eukaryotic mRNA don't have this sequence. We have something that's similar, but the one in bacteria is called the Shine-Dalgarno sequence. Now, the Shine-Dalgarno sequence is a little bit before the actual AUG, right? So let's pretend this right here is my mRNA, right? And my AUG codon is this little plastic part of the pen. My small subunit is going to bind to the initial part of my mRNA, right? Via the Shine-Dalgarno sequence, which is basically this consensus sequence that has been maintained in bacteria throughout the ages of time. It's 6 ribonucleotides, AGG. So, a double, like these triple nucleotides. Hold on, guys. I'm sorry, it's better than pausing. I take too long. Okay, so it binds to the Shine-Dalgarno sequence, and then from here, it starts reading the actual mRNA. and then until like reads it until plop, remember the little plastic thingy was AUG, it gets to that start codon. Okay, and then once we get to that start codon, then other things can occur. Now remember how I said we need certain initiation factors to help stabilize and enhance the affinity of things binding to each other. One initiation factor is called IF1, and this is actually the one that you need, the small subunit. So this, the 16S with the 16S RNA subunit requires the initiation factor 1 in order to bind to the mRNA. Okay. In addition, we also have the initiation factor 3, and this basically stops the small subunit at this point to bind to the large subunit, because at this point we still we've just discovered the AUG, and so we have to do a couple other things in order for the complete machinery, that complete snowman effect can come into play. Okay. In addition, the IF1, I'll talk about basically the ribosome's structure in just a little bit, but the IF1 is positioned in such a way that it prevents tRNAs from binding. Okay, so any tRNA that is not necessary for the AUG, that has the anticodon for the AUG and is therefore bringing that initial methionine, we're talking about bacteria here, so it's going to be the initial formyl methionine, is not going to be, it's going to be prevented by the initiation factor 1. Okay, the initiation factor 2 is actually a GTPase. This means that it can hydrolyze GTP, and it can basically start, energize basically this process. So the initiation factor 2 interacts with mRNA and the charged tRNA. This charged tRNA is the one that actually brings that first amino acid, the one with the anticodon to the AUG, right? So this one right here, so it has UAC, you can see that. Okay, this is the first charged amino acid, tRNA. You can't really see the amino acid. It's really teeny tiny. It's that little dot right there. That's the amino acid, okay, that it has. And once it actually binds to tRNA and the mRNA, basically stabilizes that structure just a second, and this sets the reading frame. So now the ribosome knows that it's supposed to read the mRNA every 3 ribonucleotides. Basically, it's going to move the mRNA continuously to the left, right, every 3 nucleotides, ribonucleotides. At this point, the IF3 releases. Remember, the initiation factor 3 was there to block the small subunit from binding to the large subunit. So when this releases, our full complex is made. It's known as the initiation complex. And so we have the small subunit and the large subunit. So now with the large and small subunit coming together, we can start seeing some of the structures, like the snowman structure that they make. And you can even see these different slots that the ribosome, that the monosome, right, has. It has the A site, the P site, and then this right here, which isn't labeled, is called the E site. And these sites are really important. They all stand for different things. The A stands for the amino acyl site. I like to call it the arrival site, because this is where your tRNA arrives. The P site is a peptidyl site. This is where the peptide bond is actually made. So this is where we form that bond between the amino acids. The E site is the exit site, boots things out. Okay, boots the tRNA that is no longer holding an amino acid out. And we'll see how this process works. So at this point in the initiation complex, okay, we have our small ribosomal unit, our small subunit, which is bound to, initially, remember, it bound to the Stein-Delgarno sequence. It read the mRNA until it got to the AUG, the start codon. At that point, the appropriate tRNA that's holding the anticodon to that start codon and the formyl methionine amino acid is going to bind to the small subunit. And it's going to bind in a position where it's in the P site, okay? And then afterwards, the initiation factors kind of dissociate. All these initiation factors are necessary for either stabilizing or just giving the actual complex time before it comes together with other things, with other bigger molecules. Now, since we released IF3, our big subunit, our large subunit can bind to the small subunit. And so you can see how the mRNA is placed, where the tRNA and the methionine, the amino acid it's holding, is placed. So everything is in, like, the AUG, which we've already read, which has a tRNA bound to it, is in the P site. The A site is now free to be accessed, right? It's free for another arriving amino acyl tRNA, or a tRNA that's charged with an amino acid can come in. You see that? And if you see, there's the 3 ribonucleotides after the AUG are now exposed. And after all this, the IF1 and IF2 are subsequently released. So this is basically another depiction of what happens. You see the start codon binding to your initiator tRNA that has the anticodon to the AUG, and it's bringing along with it methionine. So you can see this is eukaryotic, this is not formal methionine. And then through the action of the GTPase, if you remember, that was the IF2, eventually, like this whole thing gets stabilized and the reading frame is made. And then eventually with the removal of IF3, the large subunit comes in and we start forming these 3 slots, right? The A, the P, which is where our tRNA is in right now, and the E. So now we are ready for elongation. This is really teeny tiny. Elongation. So at this point, we are just elongating and growing the polypeptide chain. We're just adding an amino acid onto the next one. So onto this first amino acid that we have here, which is always going to be methionine, we're going to add the second amino acid, then the third, then the fourth, and the fifth. How does this actually happen? It happens through the action of the 23 sRNA. Okay, this 23 sRNA is found in the large subunit, and this is what we know now catalyzes the actual reaction of joining the amino acids together. At first, we used to think it was a protein, one of the many proteins that makes the structure of the ribosome, but now we know more and more that the proteins are just there for like actual support of the structure and fine-tuning, and not there for catalytic reasons. They're not actually making any of the bonds between the amino acids. So the 23 sRNA acts as a ribozyme. Remember, it just has that catalytic reaction. It can catalyze things. It catalyzes the peptide bond between 2 amino acids. Okay. Yeah, I'm going to, there we go. Actually, no. I'm going to move to this one right here. I feel like it's a better depiction of what's happening. Okay, so right now, actually, this is elongation has been happening for a while because you can see the amino acids. There's like 4 of them that are growing on this polypeptide chain. But essentially, this is what would happen even if you just have 1 amino acid. Okay, you still have your tRNA in the middle in the P site, and the A and the E site are empty. Remember, if we go back to our initiation complex, this is what it looks like. If the tRNA is in the P site, the A and the E are empty, and you have this amino acid here. So this is going to be growing, right? So if we move on to elongation, the next in the A site, or the amino acid site, also known, I call it the arrival site just because it makes sense to me, right? But the amino acid site also makes sense because the amino acid tRNA is coming in. So the tRNA that has the anticodon to the codon that is exposed, the 3 ribonucleotides, that triplet that is exposed in the A site is going to come in, right, and bind to the actual to the actual codon. So we have that complementary bases get bound together. At this point, you can see here, the amino acid that this tRNA, this charged tRNA brings with it is going to be in close proximity to the growing polypeptide chain that's found in the P site, that's held basically in place by the tRNA in the P site. So here, the 23S rRNA, remember that's the catalyzing part of the ribosome, that's the ribozyme. That's going to cause a bond between this incoming amino acid and the amino acid that was there before. It's going to make a bond. And what happens is you would think that this amino acid just gets added to the P site. That's actually not what happens. This whole chain, right? This whole chain, or however many amino acids we have here, get moved and added to the tRNA in the A site. So if you see here, all of the chain, because we've made that peptide bond between the amino acids through the catalytic action of 23S rRNA, get put on the tRNA that is technically in the A site. And then this is when translocation happens. Translocation, literally, they kind of like budge. I think the word is budge. They kind of budge over. And this is something that obviously requires GTP. You'd think that it's before the actual translocating action, but it's more just to maintain the structure of the ribosome. And it's just a very invasive thing, right? This thing moving inside of it. It's more just to maintain the structure. But anyways, this does require GTP. So at this point, with the hydrolysis or the breaking down of GTP, it releases energy and we have translocation. Okay, so the tRNA, if you see my mouse, that was here in the A site, now gets moved to the P site. and the tRNA that was moved, that was previously in the P site, now gets moved in the E site. The E sign stands for exit. Guess what's going to happen to that tRNA, especially since it doesn't have any amino acid bound to it? It's going to get booted off. So as this translocation occurs, we're not just moving the tRNAs, we're literally actually moving the entire mRNA. So the mRNA gets budged, kind of yanked to the left. And it gets yanked enough to expose the next three ribonucleotides, that next codon in the A site. So that the A site is now able to accept some more, another tRNA that is obviously complementary to it. And so now we have this growing amino acid chain, and then the whole process starts again. Another tRNA goes in that's complementary to the codon here and exposed in the A site. The peptide bond gets made again, right? That's through the peptidyl, the peptide, the catalysis of the 23S rRNA. This whole chain gets added to that amino acid, and then we have translocation, everything nudges to the side so that a new codon can be read on the mRNA. The tRNAs both get nudged one space over, right? So the one that was in the A site goes in the P, the one that was in the P site goes in the E site. And then once it's in the E site, it gets booted out. So it makes sense. I believe there's also a video for you guys here, this translation. So make sure you watch it on the PowerPoints. We go back to the PowerPoints and watch it because it's really it kind of animation obviously helps. Okay, this whole elongation process, it wasn't shown in what in this little diagram right here, but it does require elongation factors. The one that was really important is EF2. EF2, pretty much. Okay, so it requires elongation factors. Once the amino acid chain is about 30 amino acids long, and then it emerges from the large subunit, you can start seeing this kind of tail. The small subunit decodes the triplets present in the mRNA. That's what's holding the mRNA. Remember, the 16S of rRNA of the small subunit. And the large subunit is responsible for the peptide bond synthesis, because that's the thing that holds the 23S rRNA. Okay? And so it's basically a cycle that goes on and on and on until we get to those termination codons, or those stop, also known as nonsense codons. So we have the UAG, UAA, and UGA, okay? So once we get that in the A site, in the arrival site, you can see here I have a UGA, right? A UGA. What happens then is that we have a GTP-dependent release factor actually binds to it. And there's no tRNA that has an anticodon for one of these three stop codons. Okay, they do not specify any amino acids, so they don't specify any tRNAs. So at this point, the finished polypeptide chain is attached to the terminal tRNA in the P site, and the A site is vacant. In this vacant A site, the GTP-dependent release factor binds to it, and this is what binds in the A site instead of a normal tRNA, right? Because we don't have a tRNA that has an anticodon for any of those stop codons. And then this stimulates the hydrolysis of the polypeptide from the peptidyl tRNA, the one in the P site, which is then released from the translation complex. And so tRNA is released from the ribosome, and then as soon as the tRNA is released from the ribosome, everything pretty much dissociates into its subunits. Okay, and so now we have our beautiful polypeptide chain made. So you can see here, this is the GTP dependent factor, right? It comes in place of the A site. And so here, here we go, sorry. The dependent release factor right here, it comes in place of the stop codon, where we had the stop codon on the A site. And then once this happens, it basically catalyzes the hydrolysis of the polypeptide chain. It breaks it from the tRNA. The tRNA gets booted to the E site and it comes out, and then everything gets hydrolyzed. And so make sure you go over the animation, right? I'm not really sure how well the animation is going to work on Zoom, so I'm not going to try it out, but make sure you go over the animation at home. Another thing that we have to talk about is that usually one mRNA is not worked on by one ribosome. We want to kind of mRNA have a very limited amount of lifetime. They don't live for a long time. They get degraded quite quickly. So once you have an mRNA, you want to read it as many times as possible and translate it as quickly and as often as possible to make sure you get the amount of protein that you need. So usually, again, our cell just needs more than one polypeptide chain. And so what we do is we use multiple ribosomes to basically translate one single strand of mRNA. And so when we do this simultaneously, which happens all the time, we form a polyribosome or a polysome, because there's just many there's many polyribosomes basically translating the mRNA all in one go. And you can see this electron micrograph. You can see the blue are the ribosomes, the green are the polypeptide chains that are growing out, and then the red is the mRNA. It's pretty cool. So as the mRNA passes through one ribosome, its free end associates with another small subunit, forms the initiation complex, starts elongation until we get to termination again. So the polyribosomes enable a cell to make many copies of the polypeptide very quickly, and the multiple mRNA transcripts from a gene also increase the number of polypeptide chains. So this is just one of the ways that we get lots of proteins. So this is section 13.3, and I'm not going to go into too much detail about this. This just kind of shows like the different studies, talks about the different studies that have revealed the various structures and functions of the ribosome. So how we figured out that it was indeed the 23S rRNA that catalyzed things, right? All this stuff. Usually there was a lot of basically X-ray diffraction, which is how the type of analysis that was used to figure out DNA structure, right? Because ribosomes can crystallize, so that was a positive. So the individual ribosome uses were crystallized, and then the crystallized structure then showed the association of like the 70S, so the full, like, you know, snowman structure, the large and the small, with the associated mRNA and tRNA could be visualized, and you can see it at an atomic level. You can see things, the study showed that the shape of the ribosomes changes during different functional states. This attests to the dynamic translation process. As you can see, the translation is a very dynamic process. A lot of things are moving, a lot of things are happening. The catalytic steps that join together the amino acids during the translation occur under the direction of RNA, specifically the 23S rRNA in the large subunit. Yeah, so it was seen because like this was known, and like basically evidence was seen because the interface of the ribosomal subunit where the amino polymerization occurred is composed almost exclusively of rRNA and not of protein, right? I'm trying to move this down. And not a protein. And again, they just saw the proteins more as there just for structure and support and just as like fine tuning. They're found mainly in the periphery. All three sites, the A site, the P site, and the E sites were identified in X-ray diffraction studies. Again, in each case, the rRNA makes direct contact with the tRNA. And then they also found, this is actually really interesting, they found the 16S rRNA, the one that actually is attached to the mRNA and it's kind of like dragging it along, that once it drags it along and the tRNA with its anticodon is attached to it, the S16S rRNA actually releases, like it flips out two bases to check the first two bases of the codon/anticodon interaction and make sure, like look for base specificity, make sure that it's correct. So checking for accuracy. This really fits the wobble hypothesis because it only checks the first two and not the third. And so there's a lot more leniency in the third with what it can bind, right? So it really kind of fits the wobble hypothesis where there's less stringency around the third nucleotide. So that's it for this video, and I'll see you guys later.